Good morning, everybody. It's my pleasure today to introduce our lecture. This is Dr. Jana Hartmann from Technical University of Munich. She works in the group of Professor Arthur Konnerth, who is also with us today. And Jana will present her data obtained in Konnerth's laboratory. Jana really, during her scientific career, changed a lot of countries. She knows a lot of languages, but quickly she graduated from Free University of Berlin. Then she's got her PhD at Max Planck Institute for Medical Research in Heidelberg. And after that, she moved to Max Delbrück Center for Molecular Medicine in Berlin to Professor Helmut Kettenmann department. And this is exactly the place where I met Jana first time. Because I made my internship there in 1996, I guess. At the beginning of 2000s, Jana moved to Technical University of Munich, and so till now she's there, but right now she's moving to University College London, and I wish Jana you success at new place. So, Jana, Zoom is yours.
Thank you, Nana, very much for this very kind introduction. And I'm happy and very much honored to be able to present recent data from our group from the Institute of Neuroscience in Munich. At this renowned institution, although only virtually, which is a great pity, but still, it's a great honor for me. Thank you. So in my years in the laboratory in the Institute of Arthur Konnerth, I was mainly researching mechanisms of neuronal calcium homeostasis. So calcium ions are the most universal intracellular signaling molecule with a especially important role in neurons. So this calcium signaling system relies on the existence of intracellular compartments that handle calcium ions very differently, and on steep calcium concentration gradients across membranes in the cell and across the plasma membrane. The calcium concentration is lowest in the cytosol, where it is in the order of magnitude of 30 to 100 nanomolar. And calcium, cytosolic calcium signals arise, because of calcium influx from the extracellular space, where the calcium concentration is 4 orders of magnitude higher than in the cytosol, through calcium permeable channels, which may be voltage gated, ligand gated, gated by intracellular cascades like the transient receptor potential channels, or through Orait channels, on which I will concentrate in my presentation today. A second pathway for the increase of the cytosolic calcium concentration is release of calcium ions from the endoplasmic reticulum, which serves as a calcium store with the calcium concentrations between 200 micromolar up to 1 millimolar. And besides unspecific leak channels, there are 2 types of calcium permeable channels that are responsible for that: the IP3 receptors and ryanodine receptor channels that are gated by calcium ions themselves. Although the term or a tight regulation of calcium homeostasis is a prerequisite for a precise functioning of presynapses and postsynaptic sites, and although the term calcium homeostasis relates to all compartments or encompasses all compartments, in the field of calcium signaling research, it is most often used synonymously for the mechanism of calcium store replenishment. And this happens by a mechanism that is called store-operated calcium entry. And the mechanism of store-operated calcium entry was first identified in non-electrically excitable immune cells. And it was found that it requires an active complex of stromal interaction molecules, STIM proteins, and Orai channels in the plasma membrane. And there are 3 Orai homologs and 2 STIM homologs, and the interaction between Orai1 and STIM1 is the best characterized interaction of them. So stromal interaction molecule 1, or STIM1, was first identified in a genetics screen for B cell activating molecules. It is a single transmembrane domain protein that is located primarily in the plasma, in the membrane of the ER, where it functions as a calcium sensor for the filling state of the ER calcium store, and through an E-afferent calcium binding site. It binds to Orait channels that are located in the plasma membrane. So at rest, the calcium binding sites of STIM1 are occupied with calcium ions. When calcium is released, for instance, through IP3 receptors, the calcium concentration in the lumen of the ER lowers. STIM1 proteins cluster and are being translocated to ER plasma membrane junctions, where they directly bind to Orait channels in the plasma membrane, thereby gating them. Calcium flows into the cell and is taken up into the lumen of the ER by sarcoendoplasmic reticulum calcium ATPases, SERCA pumps. The ER store is refilled. Calcium binds to the EF calcium binding sites of the STIM1 molecule, and STIM and Orai return into their resting states. So the mechanism of store-operated calcium entry, or calcium homeostasis in neurons, was less well studied at the time when we began our research in this field. And also the role of STIM2 and the other 2 Orai homologs was less well known and is still much not so well researched as Orai1 STIM1, as the Orai1 STIM1 interaction. So the two types of neurons which we on which we conducted our research of calcium homeostasis mechanisms are Purkinje cells in the cerebellum and Ca1 pyramidal neurons in the hippocampus. So Purkinje cells in the cerebellar cortex are the major or the principal cells of the cerebellar cortex. They impress with their ramified extended dendritic tree that is located in the plane, in one plane in the molecular layer, where they dendrites receive hundreds of thousands of glutamatergic excitatory synapses from parallel fibers that are the axons of granule cells that are innervated by mossy fibers from the pons. And there is a second excitatory input onto Purkinje cells from climbing fibers in the inferior olive in the brainstem. The axons of Purkinje cells are the sole output of the cerebellar cortex and end in GABAergic inhibitory synapses on deep cerebellar nuclei neurons. The second type of neuron is, as I mentioned, the Ca1 pyramidal neurons, probably the most extensively studied neuronal cell type in the brain. It receives efferents from Schaffer collaterals from the Ca3 region and projects to the subiculum and the entorhinal cortex. And with regard to players of the calcium signaling and especially ER-associated calcium signaling, these cell types have some similarities. They highly express ryanodine receptors, although Purkinje cells are endowed more with the muscle type ryanodine receptor type 1, and Ca1 pyramidal neurons express the cardiac type ryanodine receptor 2. Both, however, highly express IP3 receptor type 1. And the receptors that lead to IP3 production are metabotropic glutamate receptors, that differ from the pore-forming ionotropic glutamate receptors. They are characterized by 7 transmembrane domains in the membrane and exist as homodimers. That is why you see 14 transmembrane domains in this image. And they are activated by intracellular cascades. And based on amino acid homology and pharmacological properties and downstream signaling cascades, they can be placed into 3 groups. All the identified to date 8 mGluR genes, with mGluR1 and mGluR5 constituting the group 1 mGluRs that activate GQ proteins, the phospholipase C, and finally that finally produces IP3 by cleavage of phosphatidylinositol bisphosphate. The other mGluRs inhibit the adenylyl cyclase. So I will first characterize mechanisms of calcium homeostasis in cerebellar Purkinje cells and later go on with our findings in CA1 pyramidal neurons. So we studied glutamatergic synaptic transmission and mGluR-dependent calcium signaling in sagittal slices from the cerebellar vermis, where the dendritic tree of Purkinje cells is located in the plane of the slice, and parallel fibers run perpendicularly to the plane of the slice. We whole cell patch clamp the Purkinje cells and fill them through the patch pipette with a calcium-sensitive dye. Repetitive electrical stimulation of parallel fibers leads to a release of the transmitter glutamate from the presynaptic terminals that are preserved in the slice. And a part of the postsynaptic response then is a local rise in the dendritic calcium concentration that is highlighted with false colors here. The images are the result of confocal imaging that we perform concomitantly with the electrophysiological recording. This is the time course of the calcium transient here, and in the electrophysiological recording, we, under these conditions, record a biphasic excitatory postsynaptic current, or EPSC, with a fast component and a slow EPSC with a peak around 500 milliseconds after stimulation. Both the calcium transient and the slow EPSC are sensitive to the blockade with the group 1 mGluR specific antagonist CP-CCOAT, indicating that they are mediated by mGluR1. And so fast, this means that fast or high frequency stimulation of parallel fiber results in 2 types of signals in Purkinje cells, calcium transient and a slow EPSC. Besides mGluR1, parallel fiber synapses of Purkinje cells possess AMPA receptors. They are devoid of NMDA receptors, at least in mature animals. And the sodium influx through the AMPA receptor channel appears as the fast EPSC. Binding of glutamate to mGluR1 activates the GQ phospholipase C-dependent pathway that in the end produces the calcium transient by releasing calcium through the IP3 receptor. And through a not yet fully identified mechanism downstream of mGluR1, there occurs also the activation of the transient receptor potential channel family member TRPC3. This we have found out earlier by characterizing a general TRPC3 knockout mouse, in which we performed repetitive whole cell patch clamp recordings with repetitive parallel fiber stimulation with a high concentration of AMPA receptor blockers that cancels the fast component, and also with a local agonist application with the agonist of group 1 mGluRs, DHPG. And we found that both the synaptically evoked EPSC as well as the agonist evoked inward current are absent in the absence of TRPC3. The calcium signaling, however, calcium signals, mGluR1 dependent calcium signals remained completely intact. This is important to note here, indicating that TRPC3 has no role in calcium store regulation in Purkinje cells. So we asked ourselves that maybe, or we hypothesized that if TRPCs do not contribute to calcium store replenishment, as has been proposed in other cell types, then maybe Purkinje cells rely on a similar mechanism for calcium store replenishment as non-excitable cells. And this we've started to investigate in a novel generated in our laboratories Purkinje cell type specific STIM1 knockout mice, in which we again performed whole cell patch clamp experiments in acute cerebellar slices with repetitive parallel fiber stimulation with a lower concentration of CNQX that preserves some of the fast AMPA receptor dependent component, and with local DHPG application. And we found that all components of mGluR1 dependent signals were abolished in the absence of the stromal interaction molecule 1. So this was not very surprising, or went along with our expectations for the calcium signals. What was surprising at the time was that also TRPC3 activation downstream of mGluR1 was abolished. So there was no known link between STIM1 and TRPC3 at the time, and yeah, this was a surprise for us initially. So we hypothesized, or the expectation was that most likely calcium signals disappear or are absent in the STIM1 knockout mice because the stores are empty. The other alternative would have been that the transduction cascade downstream of the mGluR1 could have been interrupted by the absence of STIM1. We tested this hypothesis by using IP3 uncaging. For that, we included caged IP3 into our pipette solution. And for achieving a locally restricted uncaging of IP3 in the cytosol of the Purkinje cell dendrite, we used a so-called lensed fiber that focuses the transmitted UV light to a spot with a certain diameter at a certain working distance. And under these conditions, a 10 millisecond UV pulse was sufficient to release, to produce robust calcium release signals in the wild type with spatial dimensions that are comparable to those observed with synaptic stimulation. And in the STIM1, Purkinje cell type STIM1 knockout mice, only 1 out of 49 tested dendritic subregions delivered a signal in response to IP3 uncaging. So the IP3 sensitive store in the absence of STIM1 and cerebellar Purkinje cells is indeed empty. And we performed another test of store functionality by locally applying caffeine, an agonist of ryanodine receptors, to Purkinje cell dendrites, and found a large reduction, although not a complete abolishment like for the IP3 dependent signals in the absence of STIM1 in these mice. Thus, it can be said with some confidence that STIM1 in cerebellar Purkinje cells regulates the ER calcium stores. And we performed later on additional experiments using a Purkinje cell type specific STIM2. So it's really STIM1 that is responsible for store regulation in Purkinje cells. So the initial expectation was then that this happens probably due to interaction with Orai1, similarly to non-excitable cells. And we tested this hypothesis in Purkinje cell type specific Orai1 knockout mice with synaptic stimulation of mGluR1 dependent responses, or glutamatergic responses, and with local application of the mGluR1 agonist DHPG. The four numbers 1 to 4 here show the 4 types of signals that are induced by repetitive parallel fiber stimulation. The AMPA receptor dependent depolarization is large enough to open voltage gated calcium channels in the dendrite. The slow calcium release signal downstream of mGluR1, the fast AMPA receptor dependent EPSC, and the slow mGluR1 dependent CYP3 mediated EPSC. And all of these components are preserved in the absence of Orai1, surprisingly. This is true also for the agonist evoked mGluR1 dependent responses. Thus, STIM1 in Purkinje cells is not regulating stores through the interaction with Orai1 as in non-excitable cells. However, we did not give up on this negative result. Using a quantitative RT-PCR approach, we characterized the expression pattern of all 3 Orai homologs in the cerebellum and in single Purkinje cells that we harvested from acute cerebellar slices. And we found that Orai2, not Orai1 or Orai3, is the most abundant Orai representative in the cerebellum and in single Purkinje cells. And moreover, this expression pattern of Orais is not altered in the absence of STIM1 in the Purkinje cell type specific STIM1 knockout mouse. Immunohistochemistry demonstrates the presence of Orai2 in a somatodendritic compartment of Purkinje cells. We used the highly expressed calcium binding protein calbindin as a Purkinje cell type specific marker. So going on from there, we started the characterization of mGluR1 dependent synaptic signaling, first in a general Orai2 knockout mouse and later in a newly generated Purkinje cell type specific knockout mouse. The gray traces here represent single recordings resulting from repetitive parallel fiber stimulation, the calcium transients, and the slow EPSC. The fast AMPA receptor dependent component is omitted here, and the black traces represent the average traces from the single recordings. And we found in both genotypes, the general knockout and the Purkinje cell type specific knockout, complete absence of all mGluR1 dependent signals. So the similarity of the hypothesis was, of course, or the conclusion, that STIM1 and Orai2 interaction is required for a normal replenishment of ER calcium stores, and this was corroborated with again with IP3 uncaging. In the absence of Orai2, there was no IP3 evoked signal, and also the cells were not responsive to caffeine application. Again, and this should be noted, that the caffeine responses, the ryanodine receptor dependent calcium release, is not completely abolished, unlike the IP3 receptor dependent responses. It is known for central mammalian neurons that empty calcium stores can be rapidly replenished by calcium influx through voltage-gated calcium channels. And we investigated the role of voltage-gated calcium channels for the regulation of ER calcium stores in Purkinje cells. We used DHPG-evoked calcium transients for monitoring the filling state of ER calcium stores. And it has been shown earlier that voltage-gated calcium channels, or the activity of voltage-gated calcium channels, can overcharge stores. We tested this by applying DHPG under controlled conditions, and immediately following depolarization evoked calcium influx through voltage-gated calcium channels. Here DHPG was applied 4 seconds after this depolarization, and the supercharging of the store is indicated by the increase of the amplitude of the DHPG IP3 receptor mediated calcium transient here. In the Purkinje cell type specific STIM1 knockout mouse, the depolarization induces calcium transient that is very much comparable to this in wild type, indicating an undisturbed signaling through voltage-gated calcium channels. And interestingly, under these conditions, DHPG delivered a calcium transient under conditions where this was not possible under control conditions in the absence of STIM1. However, this effect of the depolarization is not long lasting. It's mostly gone when the stimulation is repeated after 5 minutes. And what was surprising at the time and quite unexpected, that this transient, that the depolarization in the STIM1 knockout mouse rescues also transiently the TRPC3 activation downstream of mGluR1. Apparently, TRPC3 is not gated directly by STIM1, but it's rather regulated by intracellular calcium and not part of this presentation. And the similarity between the cellular phenotypes between STIM1 knockout mice and Orai2 knockout mice is once more corroborated by the finding that the transient rescue of mGluR1 responsiveness is occurring also in the absence of Orai2 when mGluR receptors are stimulated immediately following a depolarization evoked calcium influx. And it's therefore justified to assume that the STIM1 Orai2 complex, that STIM1 and Orai2 have complementary functions in a STIM1 Orai2 complex for the regulation of calcium stores in Purkinje cells. And that is how we envision the regulation of calcium stores in cerebellar Purkinje cells. At resting membrane potential, the interaction of STIM2 and STIM1 and Orai2 ensures normal refilling of calcium stores by the Circa pumps, and also a normal activability of TRPC3 downstream of the mGluR1 through a yet unidentified calcium binding component that may be a calcium binding molecule or a calcium dependent reaction. During neuronal activity, however, voltage-gated calcium channels contribute to calcium store replenishment, and they also can substitute for STIM1 Orai2 when the complex is absent or not functional. Our experiments have demonstrated for the first time a very close association between glutamatergic synaptic transmission and calcium homeostasis by the interaction between STIM, Orai, TRPC3 and voltage-gated calcium channels. So how does this all look like in CA1 pyramidal neurons? We started out with a characterization of these mechanisms using RT-PCR mRNA screening for the 3 Orai homologs in excised CA1 tissue and in single pyramidal neurons harvested again from acute hippocampal slices, and we found transcripts for all 3 Orai homologs. These traces here are rising curves, so-called rising curves of the fluorescence of cybergreen, that is a reporter for double-stranded DNA that shows increases of cDNA during the amplification of the PCR product. And the the curve for Orai2 rises with lower number of cycles because Orai2 is the most abundant Orai representative in CA1 pyramidal neurons, similarly to cerebellar Purkinje cells. For immunohistochemistry, we made use of the fact that in the general Orai2 deficient mouse, the protein coding exons are replaced with the LAC-Z reporter, and thus beta-galactosidase staining yields a very bright signal in the somatodendritic compartment of CA1 pyramidal neurons, indicating the expression of the deletion of the allele in the Orai2 knockout mouse, and also its presence in the wild type. So in contrast to Purkinje cells, where group 1 mGluR or mGluR1 dependent signaling was tested in the dendrites, in CA1 pyramidal neurons, we concentrated on the somata. We whole cell patch clamped CA1 pyramidal neurons in hippocampal slices and applied the group 1 agonist DHPG to the somata. This in the wild type evoked large calcium transients that were robust when the stimulation was, when the interstimulus interval between stimulations was in the range of minutes. And very similar to cerebellar Purkinje cells, group 1 mGluR signaling, calcium signaling was absent in the absence of Orai2. Although CA1 pyramidal neurons express ChIP-Cs, they are not as easily to be activated as in cerebellar Purkinje cells, and there was no current, inward current recorded under these conditions here, and therefore I omitted the current traces from the plots here. And we performed a similar experiment or an analogous experiment also in the absence of Orai1 in a CA1 specific Orai1 knockout mouse, and found that again, similarly to cerebellar Purkinje cells, the mGluR1 dependent calcium release transients are preserved in the absence of Orai1. So, so far, CA1 pyramidal neurons behave very similarly to cerebellar Purkinje cells. However, this picture changed when we applied, or when we performed similar experiments in the CA1 specific STIM1 knockout mice. Again, DHPG was applied to whole cell patch clamped somata in hippocampal slices, and the resultant IP3 receptor mediated calcium transients were found to be preserved in the absence of STIM1. Thus, with regard to the regulation of calcium stores, CA1 pyramidal neurons appear to be different from cerebellar Purkinje cells. And it turned out that they differ from cerebellar Purkinje cells in major other ways too. This was found in the Orai2 knockout mice, when caffeine was applied to the somata of CA1 pyramidal neurons. Where this agonist of ryanodine receptor again evoked robust calcium transients in the wild type, and this time also in the absence of Orai2. So indicating that neither STIM1 is required for the regulation of stores in CA1 pyramidal neurons, nor Orai2. And the initial possible explanation for the discrepancy between the behavior of IP3 sensitive stores and ryanodine sensitive stores could have been that IP3 sensitive stores may be regulated by Orai2 and ryanodine sensitive stores by Orai1, but alas, experiments in the CA1 pyramidal neuron specific Orai1 knockout demonstrated preservation of ryanodine receptor dependent responses in the absence of Orai1. And to our knowledge, because of the very low expression of Orai3 and also different properties of Orai3 when compared to Orai1 and Orai2, this is the first demonstration to our knowledge that of a ER calcium store that is independent of Orais. So neither Orai1 nor Orai2 are involved in the regulation of calcium, of ryanodine sensitive stores in CA1 pyramidal neurons. The apparent independence of IP3 and ryanodine sensitive stores we thoroughly tested in a line of several experiments. The first was the application of IP3 uncaging and caffeine application to the same cells in order to demonstrate that indeed ryanodine receptor or ryanodine sensitive stores can release calcium under conditions where this is not possible from IP3 sensitive stores in the Orai2 knockout mice. So the traces here demonstrate 3 rows of subsequent application of IP3 uncaging and caffeine application to the somata of whole cell patch clamped CA1 pyramidal neurons. And the summary demonstrates, both the traces and the summary demonstrate the abolishment or large reduction of responses to IP3 in comparison to the preservation of ryanodine receptor dependent responsive. To test the independence of IP3 receptor dependent calcium stores from ryanodine receptor dependent calcium stores even further, we designed an experiment in which we sequentially applied caffeine and DHPG to somata of whole cell patch clamped CA1 pyramidal neurons in the wild type. And we used the ryanodine receptor antagonist, the open channel blocker of these receptors that is known to deplete calcium stores, ryanodine sensitive calcium stores by sustained opening of the ryanodine receptor calcium release channel. And in the presence of ryanodine, we found a large reduction of caffeine responses compared to control responses without ryanodine, and a preservation or independence of various DHPG responses. IP3 receptor dependent responses were largely preserved. This is also shown in the summary here, where the responses in the presence of ryanodine were normalized to the control amplitudes. We tested the independence of IP3 and ryanodine sensitive calcium stores by testing the possible replenishment or regulation also through by voltage gated calcium channels. So it was known from earlier work in our group, actually from Olga Garashuk's work, the ryanodine sensitive stores in the CA1 pyramidal neurons can be replenished or even overcharged by the activity of voltage gated calcium channels. In the Orai2 knockout mice, we locally applied caffeine under controlled conditions, and 30 seconds following depolarization, the polarizing pulse to 0 millivolt that is accompanied by a large calcium influx through voltage-gated calcium channels. And the overcharging of the store was indicated by a significant increase of the resulting caffeine-induced calcium transient. However, when DHPG was used instead of caffeine, depolarization and activation of voltage-gated calcium channel is not sufficient to produce a calcium transient following DHPG application. Thus, IP3 sensitive stores do not take up calcium that enters the cell through voltage gated calcium channels. And that is also a big difference to cerebellar Purkinje neurons that we found. And last but not least, we investigated the regulation of the intracellular stores, ER calcium stores, IP3 and ryanodine sensitive by voltage gated calcium channels. First, we confirmed earlier findings for the broadband calcium channel antagonist D600 that refilling of ryanodine sensitive stores is not possible under conditions when activity of voltage gated calcium channels is blocked. We applied caffeine to single somata of bulk loaded CA1 pyramidal neurons. So the neurons here were filled or were stained with an AM dye that was applied to the entire slice. And in the presence of a calcium channel blocker, when the application occurred at short intervals, this rapidly depletes the ryanodine sensitive calcium store, and the replenishment is not possible in the presence of the calcium channel blocker, even after minute-long breaks in the stimulation sequences. This is different when after complete washout of the calcium channel blocker. There's a difference again to the IP3 sensitive calcium stores when the same analogous experiment is performed using DHPG application. Again, with short intervals between applications, the store is depleted, but when the interval is between subsequent stimulation is minutes long, the store is able to replenish in the presence of the calcium channel blocker, and is only marginally reduced compared to the condition after complete washout of the calcium channel block antagonist. So to summarize our findings in the CA1 pyramidal neurons, we have found that IP3 sensitive stores and ryanodine sensitive stores are functionally distinct and almost completely mutually exclusive. This was very much surprising given the fact that CA1 pyramidal neurons are really the most extensively studied neuronal cellular cell type in the brain, in the entire brain, I would say. And because our finding that IP3 sensitive stores do not take up calcium ions entering the cells through voltage gated calcium channels, and ryanodine sensitive stores do not take up calcium ions entering through Orait channels, we hypothesize that there is, or we assume that there is a spatial separation or the fusional barrier between two types of plasma membrane and a plasmatic reticulum membrane junctions. One that contains mGluR15, Orait channels, STIM2, and IP3 receptors, and the other one where ryanodine receptors associated with yet unidentified voltage gated calcium channels. And I would like to end my presentation with a comparison of calcium homeostasis regulation mechanisms in Purkinje cells and CA1 pyramidal neurons. What they have in common is that IP3 sensitive stores are filled by Orai2 and not by Orai1. But when it comes to the STIM proteins, there is the first big difference. In Purkinje cells, STIM1 is the major player here that activates Orai2 channels, whereas in CA1 pyramidal neurons, STIM1 is not required for the filling of IP3 sensitive stores. We hypothesize that it's possible that it is STIM2 that's doing the job here. In Purkinje cells, also ryanodine sensitive stores are filled by Orai2, which is not the case in CA1 pyramidal neurons. IP3 sensitive stores in Purkinje cells are filled or recharged by voltage gated calcium channels, which is not happening by voltage gated calcium channels in CA1 pyramidal neurons, where ryanodine sensitive stores are filled and overcharged by voltage gated calcium channels. This experiment has not yet been done in cerebellar Purkinje cells, but my strong expectation is that this would happen. Also, that this would be the case for the IP3 sensitive store in. This would also happen for the ryanodine sensitive store in cerebellar Purkinje cells, given the large overlap of the two types of stores. So as I said, the ryanodine and IP3 sensitive stores overlap in cerebellar Purkinje cells, but not in CA1 pyramidal neurons. And in cerebellar Purkinje cells, STIM1 Orai2 regulate calcium homeostasis, but not through the opening of TRPC3. That is not a store-operated calcium channel in cerebellar Purkinje cells. So thank you for your attention, and to all people involved in this work. First and foremost, of course, to the director of our institute, Arthur Konnerth. This work would not have been possible without his leadership, inspiration, and tremendous scientific input. So thanks to also the people, to my colleagues. Ryan Alexander was a master's student under my supervision, and he did a lot of work in the STIM1 knockout mice. Arjen Dijke did his PhD thesis in our group and contributed all the experiments regarding Orai2 and Orai1 in Purkinje cells. Xinjiang Chen, or Chen Engerer as is her name today, did the work in CA1 pyramidal neurons, and Rosa Maria Karl, a molecular biologist in our institute, does the PCR analysis. And I'm also extremely grateful to our many collaborators that provided us with various transgenic mouse lines, knockouts, and floxed mice, and of course to our funding organizations. Thank you very much for listening and for your attention.
Thank you, Jana. Thank you so much. Very impressive talk. So it's raised my nostalgic, you see, feeling when I was also deeply involved in this calcium homeostasis, calcium signaling. As for me, it's very, very interesting and so many new findings. Thank you so much. So we have time for questions, please. Pasha? Pasha, you can unmute yourself.
Yes, I did it, but it works not so fast as you wish. Jana, thank you for your presentation. It is, how to say, tremendous in terms of amount of data, and it is, how to say, there are so many differences between the systems. It's you did perfect presentation, but the amount of data is very, very substantial. Okay, I have several questions. First of all, you demonstrated that when you work with Purkinje neurons, you demonstrated that in Orai1 knockouts, there were no calcium responses to uncaging of IP3 in the cells, yes?
Orai2 knockouts.
Oh, Orai2, excuse me. Orai2.
Yes.
Have you checked maybe in these knockouts there are no IP3 dependent stores in dendritic tree of Purkinje neurons in general? Maybe this knockout induces, let's say, these changes. Have you checked immunocytochemically for expression of IP3 in dendritic tree of Purkinje neurons?
We have not specifically done this in the Orai2 knockout, but we have done a lot of staining for various players of ER-associated calcium signaling and mGluR1 dependent signaling in the STIM1 knockout mouse, and found that IP3 receptor distribution in the dendrites and soma of Purkinje cells is normal in the absence of a functional STIM1 Orai2 complex. So my expectation is that that would be the same basically in the Orai2 knockout too.
Okay, okay. And about CA1 neurons, you definitely, in my opinion, demonstrated that there should be some diffusion barriers between two refilling systems. It means that I think that it would be possible to check difference in calcium signaling, which, how to say, induced by activation of voltage-gated calcium channels, or system of refilling through STIM and Orai. And I would suggest that you have to see the difference in spatial distribution of calcium transients. And it should in some way overlap the zones where different system of refilling do work.
You mean on a microscopic level, there should be a spatial
system? Yes, using, let's say, just fluorescent imaging, maybe super resolution imaging, but yes.
Yes, this must be definitely done. It would be extremely interesting to pursue this. Also, in view of the fact that our finding seems to contradict the general assumption that the ER is a morphological continuum in neurons. We don't know whether this is exactly the case in CA1 pyramidal neurons, because no one has.
No, it can be continuous, but let's say different
very dynamic structure
distributed in different places.
And it may be functionally distributed, and also the ER is a very dynamic structure with constant fusion and fission events going on. So it may be possible that at least temporarily or transiently structures are discontinuous. That's also a possibility.
Okay, and the most interesting questions, these are just neurons. For me, neurons are quite similar objects, but you found a lot of difference in calcium signaling, calcium regulation. And what is, in your opinion, a functional significance of this difference? What, for what purposes the God or nature created such a difference? What will be actually the functional consequence of this tremendous by physical difference in the systems regulating of calcium concentration?
Well, that's a matter of speculation, of course. There is one interesting fact about Purkinje cells, that ryanodine receptors were not yet located in spines of Purkinje cells. So there may be a difference in the regulation of various types of plasticity. So group 1 mGluRs, mGluR1 and mGluR1/5 in CA1 pyramidal neurons are needed for, are very important, crucial for synaptic plasticity, and it's known that calcium release from stores is implicated in that, is required. So it may be assumed that there is some way of distinct regulation of these processes here, but otherwise, honestly, to be honest, yeah.
Okay,
That's,
it is good. It's good guessing, in my opinion. And in this case, maybe it is reasonable to plan experiments to see how, let's say, intervention of different systems, yes, can induce different changes in plasticity.
And there are also these minimal responses, these minimal calcium release quanta from the various release channels, sparks and quarks and whatever, and a spatial distribution in the CA1 pyramidal or a spatial distinction could have some impact here, or the distribution of calcium wave throughout the dendritic tree following stimulation at one spot, and distribution or not distribution to neighboring sites might limit or might be important for spatial for limiting spatial spread, for preventing overly spatial spreading of responses, making plasticity unspecific, for instance, or activation unspecific.
Or another possibility is that in one case you studied in Purkinje cells, you studied dendritic tree, and in CA1 neurons you studied actually regulation in the soma. Maybe, let's say, if we switch, we will see that they are the same, for example. I mean,
yeah, we have done a lot of experiments in the soma of Purkinje cells too, and there again IP3 receptor dependent signal.
It was basically
and ryanodine sensitive stores overlap in the soma too, I would say, and there's also huge store operated calcium entry in the somata of Purkinje cells that replenishes both IP3 and ryanodine sensitive stores simultaneously. So the situation in the dendrite in Purkinje cells is very much mirrored in the soma.
I see. Thank you, Jana. Thank you.
Jana, Pasha already asked even my question. I also interested in physiological aspect of your work. So maybe do you know by literature if there are any changes in behavioral of that knockout mice, especially maybe difference in full knockout and knockout in particular Purkinje neurons or cerebellar or these hippocampal neurons? Do you know such experiments, or maybe you know some diseases which related to these functional abnormalities of Orai or stem proteins receptors? I don't know.
Yes, to answer your first question, yes, we have done behavioral experiments in the STIM1 knockout mice, of course in the trypsin knockouts, and then later on in the STIM1 knockout and Orai2 knockout mice. And we found even in the cell type specific knockouts, significant alterations of motor behavior, especially on a horizontal beam that is a very that can distinguish or that detects even light changes in motor behavior, where the STIM1 knockout mice and Orai1 knockout mice were less able to walk over than their wild type litter mates. And also, yeah, so that there is some motor impairment in the STIM1 knockout and Orai2 knockout mice. And with regard to yes, and even the cell type specific Orai2 knockout mice show this impairment, although maybe on a lesser scale than the general knockout, but still there is some disturbance of motor behavior. So with regard to hippocampus related behaviors like memory and spatial orientation, we have not done experiments. So regarding pathology, there is a disturbed calcium homeostasis, of course, in many neurological diseases, especially in neurodegenerative diseases and Alzheimer's. And it was shown by Birzemjane group that STIM2 is very important in Alzheimer's. Birzemjane. Sorry, I excuse. Birzemjane is a little bit,
yeah, very close. You know Russian, very close word.
Birzemjane, yes. That STIM1 is important for the maintenance of mushroom spines in CA1 STIM2 is important for the maintenance of mushroom spines in CA1 pyramidal neurons, and it is its expression is disturbed in Alzheimer mouse models, and this leads to disturbed spine morphology and spine turnover and synaptic plasticity and disturbed calcium signaling. So all these processes are very important for normal functioning of the central nervous system and are disturbed in diseases, yes.
Thank you, very interesting, very. So more questions? I don't see the raises hands, but yeah, if somebody wants, can just unmute microphone. So no more questions. Jana, thank you so much. Well, let me check chat. Maybe here some questions. I see Lena's question. Can you read in the chat the question from Yelena Savtenko, Jana?
Let me maybe if I stop my screen. Yes, there is a question in the chat. How do you explain? Are there any doubts that in CA1 pyramidal neurons the ER is a continuous network? Well, we don't know. I don't know. Maybe it's difficult to say, because, as I said, it's a dynamic structure. Is it possible to explain that by different distance between Orai channels and Ca pumps in different types of neurons? No, I would not. I would not guess at that, because we have shown or using blockers of Ca pumps, so we know that both types of stores rely on the uptake of calcium ions by Ca pumps. So there is no difference in the dependence on Cas. Both need the Cas for the filling. Are ryanodine receptors and IP3 highly localized? Yes, both types of neurons highly express ryanodine receptors and IP3 receptors, and especially Purkinje cells. The expression of IP3 receptor 1 is excessive there. No, but also CA1 pyramidal neurons. But there is a difference in the types of ryanodine receptors. It's the muscle type in the type 1 in the Purkinje neurons, and cardiac type 2 in CA1 pyramidal neurons.
By the way, I also found very long time ago that in granular cells of Purkinje, of cerebellum, there were also some differences in initiation of these ryanodine stores and IP3 stores. They also, as far as I remember, were separated. It was not one continuous store. Maybe this is a, yeah,
maybe, yes. I should look this up, yes. Interesting.
Okay, Jana, once again, thank you so much. Thank you for your time. And we wish you the great success at your new place. And of course, to find the answers of all unanswered questions in your science. Thank you. So we should...
Thank you for giving me the opportunity.
Yeah.
And Nana, thank you. Thank you all for your attention.
I hope we will see somehow offline very soon, at least next year will bring us such an opportunity.
I will be looking forward. Bye.